Hi, my name is Dylan Butler, and I will be explaining the effect of meiotic recombination and gametogenesis of parents on predicting offspring with Punnett squares. First of all, I will be explaining to you some key terms in detail. Our first term is gametogenesis, which is the development and production of the male and female germ cells required to form a new individual. Now, gametogenesis is quite similar and follows the same pattern of division for both males and females, in which the cells both divide into two twice. Some of the differences between gametogenesis in males and females are that the cell that is divided for males is known as a spermatocyte, while the cells for females is known as the oocyte. Now, each of these cells start off as the primary cell, but they didn't divide into two. These divided cells are known as the secondary spermatocytes and oocytes. Now, here's where their paths diverge. When the secondary cells split in males, all four cells will become spermatids and they will be ready for the next stage of their life cycle. But in females, there will only be one mature ovum in three polar bodies. Now, what are polar bodies? Polar bodies are the byproducts of the segregation of the secondary oocytes, and they won't really matter anymore as they do not serve a purpose. Okay, time for our next term. Our next term is meiotic recombination, and meiotic recombination occurs when two molecules of DNA exchange pieces of their genetic material with each other. One of the most notable examples of recombination takes place during meiosis, and we will be showing what occurs during phase 2 of meiosis. In phase 2 of meiosis, meiotic recombination is used to segregate the cells containing the alleles in half, creating two haploid cells from each diploid cell. These four new haploid cells each contain their own type of trait, and then one haploid cell from one original cell will be combined with another haploid cell of the other original cell in order to create a new diploid cell that may contain different combinations of the alleles that were from the originals. If you are wondering about the two leftover haploid cells, they are just thrown away as they are non-recombinant. To show the effect of meiotic recombination on predicting offspring with Punnett squares, we have decided to pick two examples of meiotic recombination in humans, and those examples consist of hair color and eye color. Our first example is hair color, and the two alleles we have chosen are brown and blonde, with the brown being dominant and the blonde being recessive. We start off with two diploid cells, with one diploid cell being heterozygous and homozygous dominant, and the other cell is heterozygous and homozygous recessive. Using two Punnett squares, we can see that the alleles are being recombined to form different possibilities, and this leads to the splitting of the DNA. In both Punnett squares, the results are around 50-50 for the hair color the sperm or egg shall hold. Now, from each Punnett square, one result is taken, and now we are left with heterozygous and homozygous recessive. The homozygous dominant and the other heterozygous from the other Punnett squares are left over, and they are non-recombinant. Now, using a new Punnett square, we can see the results when we recombine heterozygous and homozygous recessive. Through this Punnett square, we now know that the chances for the sperm to carry a brown hair allele are 25%, for a sperm to carry mixed alleles is 50%, and for a sperm to carry a blonde allele is 25%. This is one way meiotic recombination can affect the types of alleles gametes carry. Now, on to the next example. For our next example, we are testing eye color. Now we are starting with the eye colors of brown and blue, with brown being our dominant trait and blue being our recessive trait. On one diploid cell, the alleles are heterozygous and homozygous dominant, and the alleles of the other diploid cell are homozygous recessive. In one Punnett square, it is 100% brown, and in the other square, it is 100% blue. When we go through meiotic recombination, we will be using heterozygous and homozygous recessive, and homozygous dominant and homozygous recessive will be left over. In this final Punnett square, heterozygous and homozygous recessive will be combined, and the final outcome will be a 50-50 chance to either have brown or blue eyes.